Go ahead and get started. So, welcome to STAT646, Statistical Bioinformatics. My name is Alan Dabney. I'm an associate professor here in the statistics department. I got my PhD in biostatistics in 2006 from the University of Washington, and I've been here ever since. Okay. So we'll go through the syllabus briefly. Basic info about class meeting times and locations. For those of you who are local, most of you, I think, are not statistics students, right? Not in the stat department? Stat department. Anybody else in the stat department? Stat majors? Okay. There are about as many as you following along online through our distance-based Masters in Science program. So that's what the mic is for. And so as we're talking here, everything that I say and everything that's on the screen and everything I write on the screen will be recorded and then posted to eCampus. So under class videos here, as soon as the assistants have time to process them and get them uploaded, does that usually happen, Quan, the same day? Is today's lecture most likely to be posted today, or does it happen the next today? Okay. So the day of the lecture, later in the day, under class videos, you'll have a link that you can rewatch the video if you want to. So if something was not clear or you want to revisit something that we did, you can always do that. Okay, related to that issue that there are people following along at a distance, every week on Wednesday at 6 o'clock p.m. In a different room, it'll be 411, same building, it's way down the hall, all the way down to the end of the hall and go past the stairs, is basically a virtual office hour that I hold where people can log in remotely and interact live, ask questions and stuff. Anybody's welcome to attend those if you have questions, so you don't have to. Those are also recorded, and if you can't attend but you have a question, you're more than welcome to post it to the discussion board and I'll talk about it during the Q&A. In terms of other opportunities to talk to me directly, I'm flexible. I'm usually here. I don't have a stated office hour. Just drop in if you have a question. If you don't see me, shoot me an email and we'll set something up. I'm right next door to our associate department head, and he gets lots of traffic, so I keep my door closed sometimes. So if you do come by and the door's closed, feel free to knock. I may be hiding. Okay, we have a teaching assistant, Yabu. Here's his email. And we also have what's called a technology teaching assistant who is helping with recording the lectures and then posting them to eCampus. But in terms of grading related questions, Yabu is your contact. So what is this class about? It is there's a couple of features to the class. It's about bioinformatics, and I'll try to define that in a second. But you could go to different departments on campus and take a bioinformatics class. You don't have to take a bioinformatics class necessarily from a statistics department. And part of that is because bioinformatics encapsulates lots of things. A computer science, there'd be a computer science angle to teaching bioinformatics. There might be an engineering angle to teaching bioinformatics, a mathematics angle to teaching bioinformatics, certainly a biology angle to teaching it. Generally speaking, what we mean by bioinformatics is using computational techniques to analyze and explore data that come from usually high dimensional, high throughput biological applications and experiments. So it is biological in nature, the applications. We're going to learn what is a gene, for example, what is a protein, for example, and how do I use, how do the chemists and the biologists use some fancy technology these days to get data on the presence and absence, or the activity level, relative activity levels of, say, all the proteins in a complex biological system, a person maybe, or a cow, or whatever. It's going to be high dimensional in the sense that for a sample of data that we'll play with will often look like, we'll say, n individuals that we have data on. Those might be people. So suppose we're studying colon cancer in mice. And so you do an experiment, you raise the mice under certain conditions, and you induce colon cancer maybe through exposure to something. And you, after a particular period of time, you sacrifice the mice, you extract the colon, and you pull out a piece of the tissue of the colon. So the technologies that we'll learn about allow you to extract from that sample numbers for thousands of features, which might be genes. We'll talk more about what a gene is. Might be gene-like quantities, might be proteins, might be lots of things. But for each individual, we're going to have lots, typically thousands and thousands of features. So we're going to think over and over again about our data looking like, let's say, I think the way I'm going to organize it is an M or a P by N matrix of numbers, where I have one row per feature. That might be a gene, gene 1, gene 2, ... gene 30,000. And in columns, one column per individual that we are studying. So in the mouse example, we might have 30 mice, and for each mouse we have that tissue sample, and we run it through some fancy technology, and we get 20,000 numbers for that one mouse. The first number quantifies we'll roughly say the activity level, like how active right now is gene 1. Whatever gene 1 is for this setting, is that gene doing lots right now, or is it not doing much? Is it off? And then we have a number for gene 2, gene 3, ... So it's multivariate data in that sense. And then, like, what exactly the numbers represent will vary from application to application. And what we do with those numbers will vary from, depending on what we're trying to do. There's different applications that all kind of fall within this umbrella of bioinformatics. Some applications that we will talk specifically about. The first thing we'll talk about is sequence analysis, which means let's take a biological sample and let's figure out the sequence of letters in the DNA, say. You know, if you take one of your cells and go to the right place, inside there is your DNA. And your DNA, you can think of it as a sequence of letters. And those letters constitute the code that make you up. What color are your eyes? What color is your hair? How tall are you? Do you have predisposition to cancer? Or, you know, all these different things that make you are coded for in the reference manual that is your DNA. So an interesting question is to take somebody or something and try to figure out what exactly is that person's sequence of letters. We all share lots and lots of the same letters. I share the same letters as you do. I don't remember the numbers. 90-something% of the DNA is the same between human to human. But there are important places where there are variations. That's why you don't look like I look, and so on. So in terms of understanding variation among people, what makes some people have disease and other people not, for example, it's nice to be able to look at the actual sequence of the DNA. So we'll talk about how do you deal with sequence data. We will talk about a popular bioinformatics or, quote, omic application called next generation sequencing, NGS. When I say omic, by the way, that just refers to some buzzwords that you might hear in this context. There's genomics, there's proteomics, there's metabolomics, there's lots of, quote, omics. And when people put omic at the end of a few letters, what they mean is the total collection of. Genomics would be a study of the collection of all genes in whatever we're looking at, rabbits, people, cows. Proteomics would mean we're looking at proteins, and so on. Okay. Next generation sequencing, NGS, is a sequencing technology. Sequencing has been around for a long time, trying to figure out the sequence of letters that make up somebody's DNA, or the sequence that makes up a piece of DNA, or the sequence of amino acids that make up a protein. That's not a new technology, but there's new ways to do that that allow you to do it much faster, much higher throughput, as in you can get lots more sequence information more quickly and more cheaply now than you could have dreamed 10 years ago. So you get these data that are sequence-based data, and it turns out you can use the sequence-based data, these next-gen sequencing data, to do lots of different types of questions. Like, here's 10 people with diabetes, there's 10 people who don't have diabetes. Let's do this next-gen sequencing on them, a particular flavor of it that will allow me to quantify the activity level of all of their genes. 20,000 genes for each of the diabetics, 20,000 genes, the same 20,000 genes for the healthy folks. And now let's look gene by gene and ask, is this gene different between diabetics and healthy people? Is this gene different comparing diabetics to healthy people? Most of them are not going to be different. You don't, comparing two people, say, like I said earlier, we share the vast majority of our sequence is the same because we all have to be able to breathe and shed skin cells and grow hair and all that type of stuff. You wouldn't be alive if you couldn't do fundamental things. But there will be a subset, somewhere in there there's a difference that's causing me to have DNA or diabetes and you not to. So if we have this information on 20,000 genes, we can look gene by gene and ask, where are the genes that are different? And if you can find them, and that's a statistics exercise. So this bioinformatics thing encapsulates a lot of applications. This is not a computer science class, it's not a math class, it's not a biology class. It's a statistics class, and that's what I'm trying to make it be. And that's why it's called statistical bioinformatics. Because we're going to talk about ways that you do data analysis and statistical inference with this type of data. So when the application I was just talking about, we might go gene by gene and say, here's 10 numbers, one number for each of the diabetics, and here's 10 numbers, one number for each of the healthy people, and let's do a hypothesis test, like a t-test, two-sample t-test. Are these numbers, is the mean for this group different for the mean of that group? Give me a p-value. I have 20,000 p-values now, one p-value for each of my genes. And then it might be, let's look at the subset of the genes that have the really small p-values. So out of the 20,000, we now have 100 genes that appear to be statistically significantly different between diabetics and healthy people. And then as the quantitative person, statistician, we might take that to our collaborator who knows about the biology more and say, do these 100 genes mean anything to you? And they can look at the literature and say, oh yeah, this gene, we know about that gene. That causes such and such to happen. So if it's too active, then it should be under normal conditions. We see a story that makes sense that that would be related to diabetes. But then here's some other genes that we didn't know about, and we just learned something. So we can learn something about the disease process by finding where exactly are the differences. That's one thing that we'll do. Other applications that we'll talk more about as they come, genome-wide association studies. I won't go into detail on that, we'll talk about it. Proteomics, which is looking at protein expression, protein activity levels and protein presence absence, often using mass spectrometry. Microarray-based technologies, which are getting older. We may or may not spend much time on those. And we'll also talk a little bit about what could be called machine learning / statistical learning techniques as they apply to these data. For example, my diabetic and healthy group comparison. Suppose that we could find, here's 100 genes that are different between diabetics and healthy people. Then a particular or a possible use of that information would be the next time somebody comes into the physician's clinic and is maybe exhibiting symptoms of diabetes, but we don't know for sure if they have it, can we take this type of data from them, give me a blood sample maybe, and let me get your omic data from your blood sample, and I'm going to compare your profile to cancer or to diabetic and to normal. Do you look more like diabetic or more like normal? And in that way classify or basically predict whether or not you have diabetes or prostate cancer based on a PSA measurement or something. So that's a common application in bioinformatics, trying to use these high-dimensional data to build rules that can be used, like in the clinic, to classify or predict disease, yes or no, or maybe even disease severity. Okay. In terms of background, ideally you know basic statistics like what probability distributions are, what a t-test is, what linear regression is, p-value confidence interval. Those are kind of core concepts. Those would be in classes like 651, 652. These are kind of intro classes for graduate students in other departments that we offer. If you have not had classes like those, let me know, and I can refer you to a textbook that you might want to be leafing through as we go. Because I will be assuming you know what a p-value is, for example. All right. There is a textbook. You don't have to buy it because it is available online through the A&M library website. I would start, if you haven't already bought it, I would start by just using that and see if that is sufficient for you. The only issue with this access on the A&M website is that there's a limit to how many people can be accessing it at any one time. So for example, if we all went to the A&M website, library.tamu.edu right now, and tried to pull up this book, the first 10 of us or the first 20 of us would be able to see the book. The rest of us, it would show us an error message. And that just applies, moment by moment, how many people are trying to look at it. So just be aware of that. In case you go there and you get a cryptic looking message, it may be that you may need to wait an hour. We will use R. So this R is the kind of go-to statistical software these days. Bioinformatics could look very different depending on who's teaching it. So if you went to the computer science department, for example, and took a bioinformatics class, you might see different types of software. So there's a lot of standalone software that is floating around that is specific to some of these applications that, for example, as soon as the data come out of this instrument and before it becomes the P by N matrix that I was referring to, one number for each gene for each person, it takes work to get it to that point. There has to be some processing, some substantial processing to turn the actual tissue sample through the instrument into that. For the most part, we're going to be picking up after that has been done. Usually, if you're collaborating with somebody and responsible for the statistical analysis of these types of data, somebody will give you an Excel spreadsheet or a text file or something that has the data in that format. Not all the time, but often. So just be aware that we're not going to go into all the standalone, sometimes command line, complicated software for, say, aligning genomes to a reference genome in next generation sequencing. That's not what this class is about. 30% homework, there's 7 homeworks, I think, and 2 exams. Okay. In terms of submitting homework, folks who are following along at a distance, they're in section 700. They will submit their assignment through WebAssign. You do not have access to WebAssign, so everybody else, I'm asking you to email your homework directly to the TA. So the TA's email is up here. And when you do that, please save it as a PDF, and please save it in this name format. It makes Yabu's job a lot easier. Your last name underscore your first name dot PDF. And I'm asking you to include all of your R code at the end of each homework submission, just so we can see what exactly you did to produce your output. Okay. This is my first time to do this, but I'm going to have exams use a computer. And so in the exam, download these data from eCampus and go do such and such analysis and upload a picture or insert the p-value in this text box or something like that. That's what the exam will look like. For local students, we'll be here doing the exam. There is a laptop in front of everybody. And if you have your own laptop, you're free to bring that. The only issue that I can foresee here is that we will be using R, and when you get R, I'll show you how to get R in a moment. It's free. You just go to a website, download it. It's like 30 megabytes, and open it up, you have R. But with R comes lots of core functionality to do basic statistic stuff. For things like bioinformatics, we're going to be using these packages that, for example, somebody wrote a paper about how to do next-gen sequencing-based classification using some new algorithm. And they implemented their method in an R package so that other people could use their method. It's not part of the core R distribution. So in order to get access to that, I have to, quote, install the package. And I'll show you how to do that. Some of the packages take a little while to install because they're big. So ideally, when you come to the exam, you have a computer that is ready to go. You've got the packages that you need so that you don't waste time during the exam downloading packages. I think we'll be prepared by the time the exam gets here. We'll talk more about it. I'm happy with you bringing whatever you want to the exams. So you can bring your laptop that has everything on it. You can refer to the textbook. You can refer to Wikipedia. I'm happy for it to be completely open, the exams. Okay, and I have a rough schedule in the syllabus here that we will follow for the most part. There may be some variation. All right. Any questions about the syllabus? So correlated data, I don't know that we're going to get much into correlated data. I might mention it a little bit. For the most part, we're going to be assuming independence. So if you have specific interest in correlated data, let me know, and I can try to incorporate some of that into the class. And right, so that's an interesting point that we'll talk about. Let me just come over to a whiteboard here. So I mentioned, here's an Excel spreadsheet. It contains, quote, bioinformatics data. For us, that's going to look like, call it a matrix X that is P rows by N columns. So X is this big matrix that has a number in the first row, first column, second row, first column, dot, dot, dot. Pth row, first column, X, 1, 2. There's the nth observation in the first row, nth observation in the second, and nth observation in the pth. So think of every one of those entries as a number. We'll define what those numbers mean in more detail soon. But as you go down the rows, we're talking about going across, quote, features. We'll get more detail about that soon, but that might, for example, be genes. As you go across the columns, you're going across individuals that are in your sample. So that might be the mice in our study or the people in our diabetic study. Okay. Tariq asked about correlation. Features, so gene A and gene B. They're two different locations in the DNA. I'll tell you more about what I mean by that momentarily, but there is almost certainly going to be some correlation between genes, at least some genes. So it could, because pieces or functionality of your DNA often works jointly with other pieces of the DNA. When this gene turns on, that turns on another gene, which then turns down another one, and they all kind of work together often. So strictly speaking, it wouldn't be accurate, I think, to say there is no dependence. Similarly, if you think about across the columns, what if I have 10 individuals and each individual is measured 5 times? Week 1 after exposure to something, week 2, week 3, week 4, week 5. Then there would be correlation between those repeat measurements on the same individual as well. Another similar issue is maybe like in the mouse example, we raise 10 mice and we're going to study the colon cancer issue. Each mouse, when I extract the colon, I take actually 3 tissue samples. A little chunk here, a little chunk there, a little chunk here. So now I have 3 representations of the same mouse. I might have a separate column, run each of those separately. And now I have 3 columns for the same mouse. Those are called technical replicates in the sense that they don't represent biological variability. They're not different mice drawn from the population of all mice. They are only telling us about the variation that's technical in nature. If you rerun the same mouse through your instrument, that's one kind of variation that would be there. And it is often the case that you'll have that kind of setup. I have 10 mice, and each mouse was run 3 times. In that case, those 3 columns for the same mouse are correlated as well. And we need to be careful about that. If I had 10 mice, each mouse run 3 times, I would have a p by 30 matrix of numbers, but I don't really have 30 independent samples. Just like if we wanted to know the average blood pressure of everybody in the room, we could take everybody's blood pressure and average it, or we could take my blood pressure 30 times and average that. Either way, you have 30 numbers, but mine are going to be correlated with mine. They're not really those 30 numbers for me, the variation there doesn't tell you about variation there. That's just like within person variation. That's more about what's the technical variation in the measurement of blood pressure. So just something to be aware of. We'll revisit that as we go. But when somebody gives you an Excel spreadsheet with data like these, it's important to ask that question up front. What exactly do these columns represent? Is this 30 different mice, or is it 10 mice run in triplicate? And the answer to that will dictate what you do with them. Okay, so let's go get introduced to R and start using R to learn about some basic bioinformatics stuff. Let's see. Before I do that, yeah, let's go ahead and do that. So I'm going to go to a website called, I'm just going to Google R software. And the top link will be the right one. And in order to get it, I'm just going to click on the download link. And I'll pick somewhere close. Click a link. So you can do this. You just follow the links, click it, you get an executable on a Mac, on a Windows. You can download it for a Mac as well. That's what I use. Double click it, install it, and you're good to go. When you open it, it'll look like this, a little bit simpler than this because I've already run some stuff, so there's some junk on the screen. But this is what R looks like. So R, you type in what you want to happen here and hit enter, and it runs. So 2 plus 2 equals 4. If I want to make a variable that is the numbers 1, 2, 3, 4, 5, I can do different things. You'll learn this as we go. Print that. That's now available to me as an object. X is now a variable floating around that R recognizes. X plus 1 adds 1 to all the x's. X squared squares all the x's. If I just want the second to fourth elements of x, I can subset. And in order to get started, this is not going to be terribly intensive programming class. We're not going to do a lot of R programming because we're going to use these packages that people have produced for us and use the functions in the packages for the most part. You will do some programming. In order to get some familiarity with R, my favorite resource these days is called the R Cookbook. And I can't remember if this is available through the libraries, online through the library or not. If this is a book that I would recommend buying. It's a good one. That is very simple, very easy to follow, and tells you you want to do this, then go here and I'll show you how to do it. So it doesn't start with R is built off an architecture of such and such, and this is the fundamentals of the computer science underlying it. It's practical. It's like if you want to do something, here's some example code. Best way to learn how to use something like R is to see example code and then change something. What happens when you change it? And then that way you learn what the pieces are doing. One other thing that you definitely want to be comfy with off the bat is how to get help, like documentation for something in R. There's a function, for example, that makes a histogram. So let me just make a histogram. Here's a histogram. How do I use that function called hist? If I do question mark hist, then I get documentation. And our documentation, I think, is pretty good as far as documentation goes. It will look consistent. There's a description blurb that just tells you what the function does. There's a usage blurb that tells you about all the arguments that you can use. And then there's a blurb below that that gives you more detail on those arguments. So if I want to make a histogram, the simplest way to do it is to call hist and pass in my first argument, which is some data. So over here, I'm going to make, I'm going to generate some random data from the normal distribution with mean 0 and variance 1. There's 100 numbers drawn from that distribution. And I want to make a histogram of those numbers. The simplest way to do it is just to say hist x. X is a vector or the list of values for which the histogram is desired. All these other things are optional. So if I want to customize the picture or make it pretty, add a label, all that type of stuff can be done through these. But to get started, I can make a histogram according to the documentation by just doing that. R is nice in terms of creating pictures. It's especially nice for creating pictures because anything you would like to do to modify how this picture looks, you can pretty much do it. Do you want to shade in one of the boxes? Do you want to add some text? Do you want to change the title? Do you want to customize the axis labels? All of that you can do. So I'm going to open up a text file that contains R code. And we're going to start doing some stuff in R. Under this folder called scripts, let's see. Under the folder called scripts will be text files containing R code. So I call those scripts. I recommend that as you're doing your homeworks and stuff like that, that you create text files like I'm creating here. So I'm going to open this file right here. .r, it's just a text file. So if you right-click it and say open with text editor or whatever text editor you have, you will be looking at I'm going to open, this is the text editor on this computer that I like. On my Mac, I use something called Text Wrangler. Just basic text editor is fine. So everything in here, these are R commands. If I copy that and paste it into R, it's going to do something. This, with the hash number in front of the text, is a comment. This is not code. This is just annotation. I'm writing down what I'm doing so that everybody else can read it. So let's jump in, okay? And I'm going to. I said this is not a biology class, so I'm not expecting you to have really much of any biology background. I've mentioned DNA and genes and proteins and stuff. Some of you know all about that already because you're from other disciplines. Other folks don't know anything about that stuff, so we'll be trying to balance, you know, how much detail I give. I'm not a biologist or a geneticist, so if you know more than I do and I say something wrong, feel free to correct me. But I will try to give you as much detail as you need in order to understand what we're doing without going too far into what the biology is. So to get started, let's go here. All right, so we are going to get our first exposure to an R package that has specific use for bioinformatic type things. And this will be looking at sequences. I'll tell you about sequences in a moment. Okay, so there is a repository of R packages that we'll be using a lot. It's called Bioconductor. So if I Google Bioconductor, it takes me here, and just think of it as packages that were created by researchers to implement specific methods in R. So if I want to implement the latest, greatest normalization technique for a genome-wide association study, I could come probably to Bioconductor and find the appropriate package. Then I need to install that package so that I have access to the functions. Step one. I'm going to, in order to get Bioconductor going, there's a default kind of core set of packages that Bioconductor offers that can be installed in one fell swoop. Somewhere. Okay, so to use Bioconductor, step one is to install the core stuff. And the core stuff is accessed through a function that's defined in this script. So I'm going to source this script. What source means, when I say source, is I have a text file that contains our commands. And I want to read all of that in at once. I want to run everything in this text file. I could just copy everything, paste it over to R, and click Enter. Simpler way to do that is to say, if the text file is named that, source it. So we're going to source, this particular file is actually online, so it will source it for us without us having to download it manually. This only takes a second to do. Okay. We don't yet have Bioconductor, but now we have a tool that can communicate with Bioconductor and get stuff for us. And the way I can get, I'll do this. In order to install a particular Bioconductor package, I can now call this BioC Lite function and tell it the package name that I want. The first package we're going to play with is called Biomart. So let me go ahead and run this because I think it takes a moment to download. I'm going to copy and paste. And now it's communicating with bioconductor.org. And downloading and extracting things. Okay. So it often will print out output like this. Okay, it's asking me a question. There were several packages that are involved in this, in what I just asked for, several functions and packages, and some of them have been updated since this computer last used them. And so it's asking me, would you like to go ahead and update some of these packages? It's a good idea to do that, because if the package we're downloading is going to use those, it's going to be assuming they're the latest version. So I'm going to say A for all of them and let that run. That'll take a moment. Okay. So what is Biomart, by the way? We're going to start by talking about sequences of genes. So if you just go to Wikipedia, what is a gene? First, let's ask, what is DNA? Just real briefly. DNA, like I said earlier, is the blueprint for you, or for whatever we're looking at. And it's, for our purposes, organized as a sequence of molecules that can be coded by these letters. So it looks physically like this, a double helix. And as you look piece by piece, think of it like a ladder that's curved in a helical form. If you take slices of pieces, there are distinct molecules that are joined together with these chemical bonds. There's 4 types of molecules that are joining together. There could be this molecule, there could be that one, this one, or that one. This one, you don't need to know the chemistry or anything, call it T. Call this one C. This one's G, this one's A. And it turns out that T binds to A. It wants to stick to A. And C wants to stick to G. T doesn't want to stick to G. A doesn't want to stick to C. It can happen by chance, but for the large part, if you were to read down one of these strands, if on one strand it said T, you would know what was on the other strand. Because if there's a T on one strand, there needs to be an A on the other in order for this to have this complementary nature of things. These are called nucleotides. Just jargon as far as I'm concerned for our purposes. DNA is made up of a sequence of letters. So our DNA, there's, how many nucleotides are there in the human DNA? Millions and, okay. There's like, so there's like 20,000, I'll get to that in a minute. There's lots of these letters. Lots of these letters. Okay. Why do we care about DNA, and how is it relevant? How is it relevant to, like, my eye color? Why do I have green eyes and you have blue eyes, for example? There is a part of the DNA. Take out the DNA from your cell, stretch it out, it's really long. Millions and millions of these A's, C's, T's, and G's. If you were to go to one particular area of the DNA, so forget about all those other millions and ones, let's look at this little subset, this little strip right here of A's, C's, T's, and G's. Pick that up and set it over here and let's look at it. We're going to call that a gene. A gene is just a piece of the DNA. It's a particular location in the DNA. And it controls specific characteristics of the organism. What eye color, whether the cell is going to die today, all types of things. The DNA itself, I'll finish in just a minute here, does not actually, for the most part, do anything. Think of it as an encyclopedia. It is the reference manual. What color eyes does this person have? Let's flip over to that location of the DNA. It's ACTTTJGG. They're a blue-eyed person. But those letters themselves didn't physically go make your eyes blue. In order for your eyes to be blue, your body had to read the manual and then put it into action. So think of the DNA as the reference manual, and in order for things to actually happen, the DNA has to be put into action. And usually how that happens is through molecular processes where other molecules like enzymes and things come and pull apart the DNA, take out the chunk that we're going to read right now, and make a copy of it. So if the DNA at this location for eye color said TTACG whatever, we're going to get the complementary copy of that. And now I have this little piece that's floating around that's just a snapshot. It's like a Xerox copy of the relevant page in the manual. That's not doing anything yet either, but that little strand can then be turned into something else called a protein. And a protein does do stuff. So the protein is, think of it as the product of a gene. You have your DNA. In the DNA, there's thousands of genes, separate locations that code for stuff. 20,000 something known coding genes for humans, for example. Each one of those can be, it's called transcribed or expressed, we'll say, turned into its corresponding protein product. And the protein product goes and does stuff. Kills the virus, repairs DNA damage, rebuilds the cell wall, does stuff. So diabetics versus healthy people, which genes are different is a question that makes sense because that defines, in terms of their reference manual, how they're different. We might also want to look at their proteins. Which proteins are present here in diabetic condition that aren't present in the non-diabetic condition? Because the proteins are the ones that are actually making the disease happen, most, roughly speaking. So we have DNA, we have genes, we have proteins. And there, I'll leave you with this idea. Called the central dogma of molecular biology. This is it in a really simple form. DNA, in order to be put into action, has to be copied. I want to know what my eye color is. Let's zoom into the location that houses the gene for eye color, and let's make a copy of that little section. That little copy is called RNA. It's a lot like DNA. It's sequences of letters. It's not double-stranded, and it's also mobile. DNA is stuck where it is. It's in the nucleus for you and me. RNA is free to move around and go to the manufacturing plant that will turn it into a protein. So in terms of understanding a biological system, we can look at any one of these locations. Let's sequence the DNA and look at the reference manual level, structural level differences between diabetics and healthy. We could also look at how many copies of this gene are floating around. If, for example, you have cancer, something has gone haywire. Your cells are supposed to die when they're ready. They have a life cycle. And you've got regulation going on in your DNA and all of this that tell your cells time to die. Let's pop the cell and die. If that process gets out of whack, for example, a gene is more active than it should be. It's sending out too many copies of itself, which is resulting in too many of its proteins floating around. You have overexpression of some particular process, and that can result in the disease state. So we could look at the DNA itself. What is the sequence? That doesn't tell me the whole story, though. How active is each gene? Activity would look like if a particular gene is highly active, it's out of whack, it's not in control like it's supposed to be, there's going to be lots of copies of it floating around. There's going to be a lot of RNA floating around for that gene. And similarly, there's going to be a lot of little protein molecules floating around. So one gene does not produce one DNA molecule, period. One gene, if it's needed, will turn on. And it can turn up or down, Give me lots of protein for attacking a virus right now. Or I don't need it anymore, so turn it off. Let's preserve our energy. So diabetic versus healthy. Let's look at DNA sequence. Are there structural differences? Let's look at RNA, these copies. Are there differences in activity levels of these different genes? And similarly, we could look at proteins. Is there differences in activity levels of these proteins? Okay. I don't plan to go into a whole lot more detail about biology and stuff. I'll revisit this idea of what a gene is and some of the other basic nomenclature, but don't feel like you have to go take a molecular biology class at online or something. All right, so we are out of time. What I would recommend for now is download R, get this script, and just start running through some of this stuff. Run it, take it over to R, and hit enter. Get these packages installed, because some of them do take a little bit of time to install. And we'll start going through some of this next time. Okay, questions? You do have a homework assignment that will mimic what's in here. We're doing this week, and I'm asking you to basically do similar things with a different question in the homework. So look on eCampus under assignments. We do have the first homework assignment, and it is due, I think you've got plenty of time. So it is due next Friday. And again, if you follow, as we go through that bioconductor.r script, you'll know how to do what's asked in here. All right, I will see you Friday.